Okay, everyone. So I'd like to open the last session of the 2018 Spring Symposium. So our first talk in this session will be from Ben Himes, all the way from Janelia, and he's going to come and wow us with his work on EM clarity. So Ben, the floor is yours.
All right, well, I'd like to thank you guys for having me the whole way out here, and Helen for putting up with my airline shenanigans. And I'd also like to thank, in case she's watching this somewhere, somehow, my fiancée Brenda for putting off her birthday celebrations so I could be here. So we'll have to celebrate this weekend. So I was a little bit nervous about following up all these talks. There's been a lot of very good ones, but fortunately they actually set me up and I have to go over a lot less background. So we'll kind of launch in. So this software that I've written is called EM Clarity, and it stands for Enhanced Macromolecular Classification and Alignment for High Resolution In Situ Tomography, which is about a bit of a mouthful. But what we'll do, I've broken the talk into 3 parts just to make it manageable. The first thing is sort of a forward. I'll just kind of go over sort of some of my opinions on how the field's evolving, and that'll take about 30 seconds. And then we'll get into showing you some interesting results that we've got with EM Clarity, sort of the motivation for why you'd want to use CryoStack in the first place, which I guess is my version of saying cryo-subtomogram averaging and classification. Sometimes it's cryo-STA. I just want to clarify. And then toward the end, we'll take a look at why these things, or at least why I think these things are working well, which I think is the most interesting bit, but it's also dangerous because it could eat up the rest of the talk. So that's why I put it at the end. So first off, just a little bit of a thing. So in a handful of papers that I've read about CryoStack and where it looks like it's going, kind of reads from this idea of the post-reductionist time in structural biology. And I know that we all have to sell our papers, but I think it's kind of a goofy thing to say. And I just wanted to say, I don't think it's like subtomogram averaging is going to replace SPA. I don't think SPA is going to replace crystallography. I think there's going to be a confluence of all these techniques, which we're already seeing. But it's just something in particular that annoys me, so I thought I'd point that out. So pretty much everything that I'm after here is context. And it also comes down to then this idea of how much context do you need and how much would you like. So with X-ray crystallography, we have to cage up our molecules, but we can get some very fine detail, and from that we can learn about the structure. But we can also, sometimes with some happy coincidences, a small ligand might find its way into our soaking conditions or something, where we learn a little bit something about function that we didn't expect. But we're still caged up, and there's still some restrictions that we have when we're dealing with crystals. So sort of the next degree of freedom that we can be granted is going up into single particle work, where we've got a more or less natural habitat. We can observe our molecules out in the pseudo-wild, and we can start to pick up some different ideas. So here, this little cardboard zebra, or zebra over here, I guess, is being attacked from the backside, which could be a coincidence. But if we go further out into the wild, we might lose some resolution, but we get a much better idea of the behavior of our molecules. And here we can see not only this idea of the lion attacking from the back that we saw in the zoo actually happening, we can also observe multiple molecules at the same time and realize that they can carry out different roles, even though they're the same, more or less molecule doing the same thing. They can share different tasks at different times. So observing things in the proper level of context is important for what you want to study. And CryoStack, I think, fills an important niche. So I'm going to go over, let's see, what do we have? Yeah. So EM Clarity, what is it? So it didn't start out intending to be this whole image processing pipeline, but that's kind of what it's come to be. So from beginning to end, you can take your movie frames, align them, and end up with a map. All it really needs, aside from some perseverance, is some large memory GPUs. So I'm not a great programmer, and I can use GPUs to basically make my bad code run reasonably quickly. Currently, it's available as a binary. Once the final revisions are out, I'll release the source code, and then you can hopefully make that much better for me. And then there's information available on the wiki. There's a tutorial and some YouTube videos, which if you struggle with making documentation, I highly recommend, as long as you don't have to watch them yourselves. The screencasts work great, which I had the unfortunate experience of hearing myself play back during a fortunate experience, which was a workshop that we had at EBIC earlier this week, where we had enough people interested to actually work through some of these methods that we spent 2 days playing around with subtomogram analysis, and it was pretty cool. And so one thing I did over here while we were going through this workshop was that, well, yeah, it's programmer-friendly program. So just that's my way of saying it still runs from the command line. There's some additional scripting you need to do to really get it to work. It's not something you're going to show up on day one and make happen right out of the box, but it's a lot easier than writing the programs yourself. So looking forward, we would hope to integrate this within CCPM with the help of Colin and Tom, and hopefully also bring in some additional resources like a nice maybe user interface that also gives you some good feedback and keeps you close to the data, as we've talked about in a handful of talks, being important to be close to the data in cryo so that you're not having mysterious particles appear out of nowhere. All right, so that's the end of the first part, just a few little sort of spiels. So there's motivation, so why do we want to use CryoStack? I've already sort of set this up. We can observe things in a richer context, and so the question is, what can we observe? So this first part of the name, this classification, has to do with essentially particle sorting based on compositional or conformational heterogeneity. And the results I'm showing here are 5 out of 7 classes that we got from an Empire data set. This one is about 3,000 mammalian ribosomes that have just been coarsely purified by being spun down from a rabbit reticulocyte lysate. So it's a pretty rough, we'll call it ex vivo preparation. It's certainly not in situ, but it's certainly not a clean single particle preparation either. The classes I'm not showing, this is a ribosome. The blue is the 60S large subunit, so there's an additional minor population that is just the 60S subunit, and then another small piece that's about equivalent to another 100 particles, which was just junk. So that actually brings me to the first point before we even talk about what any of these things are, is the absolute numbers. So this is a data set that has 3,000 particles in it, and while a couple different people, notably Christian Spahn and a few others, have shown that you can do some pretty cool and advanced classification, particularly with ribosomes, those data sets are 2 to 3 orders of magnitude larger. So 3,000 particles is something a little bit more tenable in terms of what you can get from not only potentially in situ samples, but also kind of more realistic ex vivo sort of situations. So aside from being able to isolate minority populations and also doing that from very small sample sizes, we also get some really cool separation. So the 2 ribosomes on the far right have a sort of mid-ratcheted small subunit. And I don't know a lot about the biology, but I had to learn a little bit coming in here, but that's one of the things that happens when this machine moves around. And they both have this translocational cofactor, elongation factor 2, which kind of boots the tRNAs through the mRNA channel and pushes the stage of translocation to the next step. We have a different cofactor in class 1, which is the elongation factor 1A, and that is partially occupied. So there's only about 900 particles in this class, so the occupancy is limited, but you can see the A site tRNA charged and coming in, as well as a P site tRNA as well. And these other 2 classes are only different by the population of an E site tRNA, which is pretty wild because that tRNA is about 25 kilodaltons in mass, whereas the total mass of the ribosome is about 4.2 megadaltons. So statistically speaking, we probably shouldn't be able to separate those populations based on that mass difference, and what that implies is that there is some degree of correlation. There's some functional state that's different that's holding that exit site tRNA in place, and I just don't know enough or care enough, honestly, to go looking. But if you had a sample you cared about, that's something that you should be able to do. Another quick example of classification, again, the ribosome, this time from an ADS yeast RNA. So this time, rather than being purified with all the components you need for translation, this is an empty, non-actively translating ribosome. And what's interesting about that is that we've got these 2 different domains. In orange, you've got ES27, which is an expansion RNA segment. That can be up here or down here, and we don't really know exactly why it swings like this. But what we've seen is that when it's in the up position, it's right by the peptide exit channel, or tunnel. And when it's down, it's pointing toward this green blob, which is called the L1 protuberance. And really just the take-home message from this slide is that these 2 domain motions, when the ribosome is not transcribing or translating, is just, they're stochastic. There's no correlated no correlation between their 2 positions like there was with the tRNA in the last slide. And that is just something that's difficult to isolate because these states are just flipping in a way that is hard to pick up. So it was pretty cool to be able to see these things come out. All right, so we'll move from the classification into the high resolution bit. So again, we're seeing on the top row, these are the original maps that were deposited by the original groups that did the studies. So the yeast on the left was published at about 13 angstroms. And in EM clarity, we get down now to about 7 angstroms resolution. The mammalian ribosome was published around 11. And we took it down to 8, but I haven't processed that since we've gone through and debugged a bunch of things, so it probably would go down lower. But I guess we don't need to really detail what's going on here. It was going with the original orientations that Reliant published. I should say Tanmay published using Reliant. And showing stepwise, using just those orientations, the improvements that you get from different features of EM clarity, which I'll detail in the third part. But I just wanted to give you the impression that these changes, 11 to 7, I think most of you realize is a pretty significant, not only in terms of magnitude, but in the range that you're moving through. It's particularly important in terms of what you can interpret from the map, moving from 13 to 7 angstroms. You get much clearer resolution of both the RNA and peptide helices here. And then something very exciting that Beata mentioned earlier is that they've just recently released this HIV data into the Empire database. And in working with that original set of tilt series now, we're at about a resolution that is comparable to what they were able to get. I don't want to make much of the numbers until we look at the full data set. But I do think it is really cool that when I started my PhD, just getting a sub-nanometer structure was really the primary goal. Florian scooped me on that about 4 or 5 months into my PhD, which was a good lesson right out of the gate. But now we can see side chains, and we can see side chains reasonably clearly. I mean, these are a little bit clunky, but I'm pretty optimistic that we'll get down close to 3 and maybe even a little bit beyond as we process the full data set, which is running right now back in Virginia. All right, so high resolution, we've got classification, but what are these all really aimed for? It's for the ability to get to a truly in situ sample. So assuming that you can thin your sample down enough, Corey mentioned earlier in his talk the Fib milling that's available, and then we also have a technique where we use a particular phage protein to pop a little hole in bacteria and gently but quickly lyse them. We can view proteins in their closest to native context. And in this case, this is an array of chemotaxis signaling receptors that are in the inner membrane of the protein. Now, this is at a pretty low resolution. This is 4 years ago. We showed that even with what was then the precursor to EM clarity, we could get down to about 18 to 20 angstrom resolution. And importantly, that soluble peptides that normally do associate with the receptors are retained during this lysis process, which was good to know. In Fib milling, that wouldn't be a problem anyhow. So we have some new data collected on new detectors with new ideas about the level of defocus to use and everything that we've learned over the past 5 or so years. And we're processing that now, and there's some pretty interesting results coming out of it. So this is the end of the middle section, so I've got another little acknowledgement thing. I'd like to thank the Emperor of Empire, Ardon, for the work that they put into maintaining this database, because although I was able to make some pretty solid strides using our own in-house data, having access to these data sets has really accelerated, just as was their goal, the development of the methods that I've had, and resulted in not only better maps, but hopefully better tools that will allow you all to get better maps as well. And then also just a quick shout out to the people that took the time and also the little bit of risk in putting their own data up there. It's well appreciated. All right, doing good on time. So we're going to take a quick look at how, and maybe a little bit into why, some of these things work. So we were set up nicely by Tim earlier, and then Julia later talking about the missing wedge effect. And what we're looking at here is just the sampling of, I don't know if it's crystal around, projections, or images, we'll say, in the Fourier domain. And there's a couple important things that we have with tomography. So what makes tomography powerful, particularly when it comes to the classification, is that we have a unique 3D structure for every particle in our sample. And that's what really makes the classification more powerful than it is in single particle, assuming that you can account for the distortions that this imperfect sampling leaves behind on your particle. So as we tilt the specimen through and collect multiple images, it accumulates radiation damage. And as it's already been mentioned, the specimen gets thicker, so your signal-to-noise ratio goes down. And that's represented here by the orange disappearing, so the high-resolution information disappearing as you get up to higher tilt. And then there's this missing region, which we've talked about already. And if Chris didn't already make it a little bit unsettling to say that the projections we collect because of the Ewald sphere aren't strictly projections of our specimen potential. I'll make it a bit worse, and I'll say they aren't even two-dimensional projections to start with. So because physicists like to abuse math a little, we sometimes ignore the fact that when we talk about the Fourier slice theorem, what we say is that the Fourier transform of a two-dimensional projection is the central section to the 3D transform of the object. But that's only true if we're integrating something that does not have a finite support, which our molecules are clearly finite in dimension. So what that means is that our projections actually have a thickness. So our 2D projections actually are more or less 3D in reciprocal space. And the reason that I took the time to mention that is that all the work in the last 15 years or so, it's been most concentrated in the last 15 anyhow, in working with subtomogram analysis is centered around not really compensating, all that terms used, but estimating the effect of the missing wedge and how you might deal with that in the alignment and classification process. One of the things I think that makes EM Clarity work well is we do a good job estimating what these effects are, and part of that is in calculating the thickness of these projections, which results in to what extent they overlapped at what resolution, and it also means there's some low resolution information that there is no missing wedge. So that's one of the things that works pretty well. So a typical cryostack workflow, we'll kind of pop through this. Each of the things in red are areas where we've implemented something that is either better or somewhat novel. But the basic idea is that you get your data. You come in, you have to determine the defocus, which Kai nicely set up, and I'll have a few things to say about that. After you determine your defocus and the relative rotations and scalings and shiftings between your projection images, you can reconstruct a three-dimensional volume, which we call a tomogram. And then where subtomogram analysis comes in is you go in and pick, either manually or with a template, your subvolumes, cut them out, and then you can align them together iteratively, which is this little loop here. And you can get pop out with a structure. And there's some more details in there. So this branch, even though I like the results a lot, I'm not going to talk about the how, because it's more than I have time for. This is about an hour-long talk. This classification, I will take just the time to say that one of the neat things that we do is that we sort of encode a priori information into the classifier that we feed to the clustering algorithm that basically tells it a little bit about the different length scales that we think are important for biological molecules. This Tomo CPR is a totally novel loop that other people have tried to do similar things, but the basic premise is that you use the information from the subvolumes like fiducial markers to come back and refine your tilt series geometry. So we actually pop out of this loop, really, come back up and reconstruct the tomograms iteratively throughout the cycle, which makes the 3D CTF correction a little bit more expensive because we have to do it multiple times. Actually, I guess why don't I say a word about that now? So the way we handle that in EM Clarity is to, Beata mentioned that the time scales really dramatically with the thickness of the slab that you reconstruct. So what we try and do is estimate at this given binning, our resolution maybe only goes to 12 angstroms, so we only need to correct maybe 3 slabs out of the whole tomogram. And as you get to higher and higher resolution, you reconstruct more and more slabs. So it's sort of trying to play that trade-off between speed and accuracy. Okay, so this is what the data looks like. These are from that HIV data set that they popped up, which I've been sneaking off to my room and working on breaks. So this is from a tilted image of 0 degrees, so a non-tilted image, and an image tilted at about 50 degrees. And you see they look more or less the same. There's more overlapping information at high tilt, and you can see there's some gold beads. They've been erased here, kind of poorly, that come into view that are not there below. So the first thing we want to try and do is determine the defocus. And generally, at least historically speaking, the best we've been able to do is determine a nominal defocus at the tilt axis and one for the whole tilt series. But what we really want to do is get a value of the defocus for each tilted projection. So on the left, what you have, and this is a 1 angstrom pixel sampling, so this fit goes out to about 5 angstroms, which is, I think, pretty good for tomography. You get a nice clean fit. You can see, 5 or 6 tone rings, depending on how much you squint. Certainly not as nice as the stuff Kai was showing, but, and then in a tilted image, you see that you get basically crud. And the reason for that, I think, has already been explained pretty clearly, so I won't dwell on it, but it's that the CTF is defocus dependent, and you basically have destructive interference as you add these things together. And what we might be able, or what you might be able to imagine, I should probably make an animation for this, is that you could possibly resample these images, though, to make the zeros happen to line up. And that's exactly what I tried to do, and it seems to work pretty well. So I'll tell you a little bit about that. So we don't know what the focus is at the tilde axis, but what we can calculate is the difference in defocus. And if this is what you're doing with the general resampling, I'll have to take a closer look at that. But we calculate the difference in defocus here, and from that we can estimate a resampling factor, which to do that, we basically just say that given where we work with tomography anyhow, the spherical aberration term that's part of this equation that we use to calculate the CTF is pretty minor compared to the defocus term, so we'll ignore it to start with. And just with some algebra, we come out with this thing that says we can either resample the pixel size or change the transform size, which is what we actually do when we take the Fourier transform to get to the power spectrum. And it turns out this is actually pretty good until you get down to close to focus imaging conditions when it becomes worse. So what we've added in is a small fitting procedure that starts here and then wiggles around a little to see what the best actual factor would be. And then what we get is something that, I promise it's not just the scaling with the contrast. I didn't have time to come back and fix this, but you can see, 4, maybe 5 tone rings here on the right from the same image that was this guy on the left. You can also see that there's actually quite a bit of astigmatism in this tilted image, and if I were to show you the images from around this one, it gets progressively worse as you get up to the higher tilts. So this is something that works pretty well, and it's pretty painless to do. You just pop it in after you do your initial CTF estimation, and you get a refined view of the defocus in the tilted images. So that's a first step toward improving things. The next is the template matching. So in EM Clarity, we don't do any very large, I guess you would be free to search a large angular space on the subtomograms, but what is much faster is just to do your large angular search on the tomogram. So that way, every time you rotate your reference, you're essentially searching against every single subvolume at the same time. So what I'm going to show you are the results from just one of these tilt series that we popped up. And in EM Clarity, there are some visual editing tools that you can manually edit these results, but in this particular case, it's accurate enough that you can choose geometrical constraints and say, I want 3 neighbors that have, or each particle must have at least 3 neighbors that are so close to each other and maybe have some acceptable level of angular deviation, which is all I did here. Which is really nice to be able to do because this data set has about 200,000 particles in it, which I wouldn't want to pick by hand or even clean up and edit by hand. Some interesting thing, we're not sure yet if these sheets of broken virus are going to be useful, but this is something that you wouldn't get without the template matching because you're looking for the full particles and then maybe building that on onto some circular lattice. You can see the curvature actually is still maintained in those sheets though, so I have a feeling they'll probably be useful. Maybe not, we can always delete them at the end, but the template matching in EM Clarity is pretty powerful. We use some interesting things that we don't have time to cover, I guess, today. So that gets us into the CryoStack workflow, and I didn't list the CTF correction there, but since Beata showed such nice results with the 3D CTF, I sweated it out and put something in EM Clarity that at least gets close to what she does. Probably a little slower, but it works pretty well. So next we get into CTF amplitude correction. So we flip to the phases, or multiplied by the CTF really is what we do as well. But that leaves large areas that aren't corrected, which is something that you typically do in single particle with a Wiener filter. And what I'm showing you is what happens if you don't take care of this. This is the raw subtomogram average from that ADS East sample. And what we see is some really cruddy averaging out in this direction, whereas here we see clearly nice alpha helices and RNA helices. And the reason for that is that the resolution is fairly anisotropic, which has been discussed already a bit earlier. You see in this one direction it's particularly bad, dropping down maybe around 12 or 13 angstroms. And then in some, it's actually much higher than the global resolution. So the idea is to be able to adjust the Wiener filter that you use for the CTF amplitude correction to take into consideration these amplitude modifications. What I did is I took this thing that Chuck Sindelar and Nico published 8 or 9 years ago. It's the volume normalized single particle Wiener filter. And there's 3 different things we had to do to make it, actually 4 to make it work well. So I'll talk about those real briefly. And I guess I'm running short on time, so I'll clip through these last couple. So the first thing you need is an estimate of the solvent content in your volume, which we do with this iterative masking routine that we have built in. We took the FSC, which is used to estimate the spectral signal-to-noise ratio, and instead of calculating that over spherical shells, we calculate it over conical sections, which have been shown before for determining the resolution, but hadn't been implemented in terms of actually correcting for it. And then the final thing that we had to do here was just in order to map the expected signal noise from your average back to the particle, which is necessary for a Wiener filter to work. This was a spherically shelled average thing in Nico's paper, so what we did is we just took basically a simple convolution to smooth it out, so that way it is also anisotropic, which you would want to be able to fully correct. And we see we get some nice results. So I have one slide for the Tomo CPR, and I'll be done. So I'm just going to show really cartoony how it works. The idea is similar to single particle, where we project our high-resolution model down, and then we compare it to the data projections. The main difference is that we take the full reconstructed tomogram, which is going to have other things like non-target particles, carbon, gold, other interesting things that we might want to look at. And then we cut out the areas where our subtomograms are and replace it with the higher signal-to-noise ratio average that we have. So we get some gain. We project that down, and then we convolve it with the appropriate CTF, depending on this defocus gradient. And then we can compare it to the raw data projections, which we then feed into IMOD as if they were gold fiducial markers. And the real big advantage of doing it this way, then, taking advantage of IMOD, is that we get local resolution estimates, or local geometrical orientation estimates, which then, when we do our reconstructions, no longer are we reconstructing a simple tomogram, but it's warped in local areas to match the actual movement of the sample, which, in particular with tomography, when you have a wide field of view, you're going to have more local motion within that field of view. So this has turned out to work really well, and it's responsible for really, I'd say probably the bulk of the improvement in EM clarity. So if you want to incorporate this in your own workflow, it's not too hard. Just go ahead and do it. I guess one other last plug is that when you're doing this, because you're applying the CTF to these tiles, you can sweep a range of CTFs around the given one and refine the defocus on a per particle level, which is something we've done. It seems to work pretty well, but I haven't tested it thoroughly enough to comment with any numbers. But it seems to be a pretty good thing. So future directions, further mergers of both the ideas from a single particle into CryoStack, but also some of the things we're talking about with Nico, we're going to take some of the ideas from tilted images and bring them into single particle, and that's why I really think there'll be a confluence of the two techniques. I hope in a couple weeks we might have a map that is down to 3 angstroms or maybe a little below. We'll have to see. And hopefully with the help of Colin and Tom, we can make EM clarity easier to use and have some better and faster feedback. So with that, we do some quick thanks for the third part of the section. I'd like to thank Pei Jun for supporting me during my PhD. Jing Ning was a lab tech in Pittsburgh who expressed many of the proteins I worked on. And Xiaofeng Fu collected a lot of the data I worked with and also was a great beta tester for many of the early versions of EM clarity. And of course, funding. And then I've been at HHMI Janelia Research Campus with Nico since July. So a special thanks to him for being patient and allowing me to continue on with this pet project a little bit. And also Tim and Peter for letting me chew their ears off with annoying questions. So with that, I will conclude.
Thank you, Ben. We have time for some quick questions.
Hi, so I had a question which is probably not quick, which is how the 3D classification actually works, but maybe I can ask you later.
Sure.
One question that might be quicker I had is, when you do the refinement of the tomogram, you should be able to trace back the deformations that have occurred, during the data collection. So do you think looking at the extent of these deformations that are really a limiting factor, for,
yes. So
and
even,
to what extent?
To what extent? So a significant one, I'll say that. So the biggest single jump and improvement in resolution is from this correction of the local distortions. So kind of a hand-wavy thing. I was just telling Beate at lunch, when I did the initial template matching so their sample has got gold beads all over the top and bottom, so it's about the best alignment you should be able to get with gold beads. I got 130,000 positive hits after I cleaned up the template matching results. I went through and did one round of this Tomo CPR and came back and redid the template matching and got 200,000. So just from that sort of hand-wavy, assessment, you should be able to pick up much better results. And then there's the resolution improvements too. So the classification I can do in like a 20-second nutshell. we take this 3D CTF and we use basically the wedge mass differences that John Heumann published a few years ago. So basically you calculate an expected value for your particle by distorting your average with each particle's 3D sampling function. And then you look at how different that distorted average is from your average particle. And that difference is what you classify on. We do that for 3 different resolution bands where we take basically what's the opposite of a notch filter, a bandpass filter, around a particular resolution, so maybe around 10 angstroms, 18 and 30 or something. Then we do PCA on each of those resolutions, concatenate those matrices, and then classify based on that.
Any more questions? Actually, I have a question. So I'm really excited about all these high-resolution tomography maps that are going to come out. And I'm looking forward to building models into them. Mm-hmm. Are there particular kind of eccentricities of these maps, or pathologies, if you like, that we should be aware of compared with SPA maps? Sure.
So I don't think that they're unique to tomography. I think it's something that actually we should be considering more with SPA maps. Actually, I think I asked Ardon about that after his talk, was the anisotropic resolution you have, not only spatially, but then directionally. I mean, that's something that happens in single particle reconstructions too, quite frequently, particularly when you look at smaller populated classes. It isn't really advertised that way, but if you flip through the EMDB, it's pretty obvious when you see these maps that are kind of like streaked out a little bit. And that has to affect the side chain densities, and it has to affect how you interpret, like, you know, if you have a phenylalanine that's smeared like this, is it actually rotated or is it just smeared? So anisotropic resolution. Yeah. Okay, if there are no more questions, I think in the interest of time,